as I said, we are going to finish up this biology lecture talking about a process known as meiosis. And meiosis is a process where a diploid cell forms gametes. And gametes, we'll talk about in a second, are haploid. So you remember the diploid means that there are homologous pairs of chromosomes. A haploid cell means that there are not homologous pairs. There's only one homolog from each chromosomal pair in each gamete. So we're going to go through the process and show how this comes about. As I said, gametes are the sex cells, or the sperm and the egg. And these are the only normally occurring haploid cells that we have in our body. All of the other cells in our body are diploid. All of the other cells have homologous pairs. The gametes, or sex cells, are the only ones that are haploid and do not have homologous pairs, only have one homolog from each chromosome. So let's look. Now, because it results in haploid cells, it's a little bit different. Now, the names of the processes in meiosis are the same, but meiosis is divided into two parts, meiosis 1 and meiosis 2. And the important part when you look at this, and we're going to do it twice so that you get an idea, is to understand that you have to go from a diploid situation to a haploid situation. So up here, when prophase begins in meiosis, you have two homologous pairs, and they pair up here, okay? And then they separate in a very similar way to what we've seen before. But what results is two cells that are no longer homologous. You start out with two pairs, and you end up in each one with just two genes. Up here, if it was a human, it would be 46, or 2 times 23, and down here it would be 23 and 23. So you end up at the end of meiosis 1 with two cells that are haploid, or have only one of the two pairs, but they are duplicated. There are two haploid cells with duplicated chromosomes. You start out with one diploid cell, you end up with two haploid cells with duplicated chromosomes. Now, meiosis 2 takes these two haploid cells with duplicated chromosomes. You see, these are still duplicated, connected at the centromere, duplicated, connected at the centromere, but one of them contains this pair and one of them contains this homolog, so they're separate from each other. And this diagram does this so you can tell these are two pairs, but they're different. And so then they line up in the same way, and these duplicated then are separate, and you end up with 4 cells, haploid cells, that do not have duplicated chromosomes. So you start out with 2 haploid cells with duplicated chromosomes, and they're split, and you end up with 4 haploid cells without duplicated chromosomes. Now, sometimes people find this a little bit confusing, so I have another diagram that puts all this in the same picture, and hopefully this will help. Now, remember, you start with interphase, and interphase is that period of time where nothing is happening, okay? But what is really happening is you're actually duplicating your DNA in here, replicating your DNA, let me say it that way. And you start out, you have 2 pairs of chromosomes that are duplicated. Okay, this is one pair here, and the duplicated chromosomes are attached at their centromere. So here's one chromosome that's been duplicated, attached to the centromere, and here's the other pair of that same homologous pair. So this whole thing is the homologous pair. This is a homologous pair, but they're also duplicated, okay? Then they go through one set of processes here, metaphase, they line up, and then the pair, the homologous pair, is split. So now you have a haploid on this side and this side, but they're still connected at their centromere, so they're still duplicated. So you end up with two chromosomes that are duplicated. But here's the one of the pairs, here's one homolog of the pair, and here's the other homolog of the pair. Then they line up again, and they're separated again, and the duplicated chromosomes are then separated. And so you end up with 4 cells that each have its own single chromosome here and single here. So for n equals 2. Again, if we do this for a human, 2n would be 46, or 2 times 23. You have duplicated chromosomes attached to the centromere. They then separate, and you end up with 2 cells that have 23 chromosomes each, but they're duplicated attached to the centromere. Then they divide again, and those duplicated chromosomes are separated from each other, and you end up with 4 cells, each of which has 23 chromosomes. Hopefully this helps, and read through the section, look at the diagrams very carefully, and I think that'll help you figure this out. Now, here's a diagram that looks at specifically at the production of the human egg. This is called oogenesis. And you start out with an oocyte that goes through the first meiosis and then the second meiosis, and you end up with 3 little jewels here called polar bodies and 1 oocyte. Well, in a male, you end up with 4 cells, all of which are about the same, and those are called sperm. And each of those result from the meiosis in a male. Now, all 4 of these work in the male. But in the female, you produce a single ovum, or oocyte, and then the rest, the other 3, become polar bodies. And these do not develop, and they basically are lost. And so for each meiotic process in a female, you start out with 1 diploid oocyte, and you end up with 1 haploid ovum that results in a baby if fertilized. Whereas in a male, you start out with a diploid cell, and you end up with 4 haploid sperm cells, that all of which function properly. Okay, so now let's move on and finish up with a short discussion on virus. The reason that virus is talked about is because of the way that virus replicates, and it replicates or reproduces itself through the process of interfering with the cell's DNA or RNA production. Now, to start out, a virus is a non-cellular infectious agent, and it has 2 distinguishing characteristics. First of all, it has either DNA or RNA in this protein code here, it cannot reproduce on its own. It cannot reproduce without having a host in which to inject its DNA in. So this shows how this happens. The virus has to attach itself to a cell, and this attachment is very important, and one of the reasons why I'm emphasizing it is because one of the ways we fight off viruses is we prevent them from attaching to the cell. Then it penetrates the membrane, and the viral DNA is then injected into the cell, and it makes its way into the nucleus. Now, this is a virus that's infecting a bacteria, and you remember bacteria are prokaryotic, so they don't have a nucleus. But basically what happens is that the viral DNA is then incorporated into the bacterial DNA, and it's replicated along with the bacterial DNA, and it co-opts the cellular mechanisms in the bacterial cell and produces new virus. Now, once this becomes loaded with virus, then a process known as lytic pathway occurs and releases the virus into the system. So you start out with penetration, replication of DNA, then synthesis of new virus, assembly of the virus, and then rupture and release of the virus to infect other cells. So how do we go about fighting this off? And this is important. There are two ways that we do this, and both of them require antibodies. Antibodies are proteins that aid in the destruction of any infectious cell. They also aid in the destruction of cancer cells and various other cells. Now, they can work essentially in two ways. The antibody can bind to the virus itself in such a way that the virus cannot attach to the cell that it would generally infect. And that's what this is showing here. Once this virus is bound, it is then neutralized, and these are called neutralizing antibodies. And this is the humoral aspect of our immunity to virus. The other way is that when the virus infects a cell, the cell is changed in such a way as it's recognized as abnormal. And then phagocytic cells, cells that engulf invaders like virus or infected cells, come in and they attack the infected cell and kill it before it can produce new virus. And so they kill the cell that's been infected. And so these are the two ways that one is called humoral immunity. This is the binding antibody against the virus. And the other is called cellular immunity. And this is when the cells, with the help of antibody, recognize an infected cell and destroy it before it can produce new virus. Now, this is the time of the year when people get their flu vaccine. So what is a vaccine? Well, a vaccine can be a dead or weakened version of any infectious agent. In this case, it would be the flu virus. And what that's injected into the animal or person, and it stimulates the production of antibody. Now, because it's weakened or dead, it doesn't cause the infection itself. It only presents the things that are necessary for the body to recognize the virus, and it produces an antibody. So that if a real infection comes along, the body says, well, wait a minute, I've seen that, calls up these antibodies and either neutralizes it or fights it off. And so that's how these vaccines work. So that finishes up our discussion on the cellular aspects of reproduction, and also ending up there with a short section on virus. And next week, we will talk about Mendelian genetics.